So this is the introductory lecture in a mini-series of lecturelets on statistics and statistical analyses of data. And of course, I'm going to be thinking mainly about time frequency-based analyses and statistics of these kinds of data. But a lot of the points that I'll mention and things that I will discuss are relevant to any kind of statistics for any kind of data. So first, this may seem like a stupid question, but I think it's important to think about why even perform statistics, and do you need to perform statistics? Do you need statistics to determine the direction of your findings? And by direction here, I'm referring to, an increase or decrease relative to the baseline, or relative, for condition A relative to condition B. Do you need statistics to determine what patterns of results are predicted by your hypothesis? Do you need statistics to determine what findings are relevant for a particular theory, or what findings are kind of unpredicted by a theory or orthogonal to a theory? Or do you need statistics to determine which findings you can interpret or not interpret? So the answer, as it turns out, is none of the first three. So you don't need statistics to determine the direction of your findings. To determine the direction of the findings, you need to plot the data. You need to visually represent the data in bar plots, scatter plots, or time frequency plots, topographical maps, whatever. For really simple data, you can just report the means. You don't even need to have any visual depiction. But statistics won't help you in determining the direction of the findings. Statistics are also not going to help you determine what patterns of findings are predicted by an hypothesis or important for a theory. Only, human intellect can answer these questions, and statistics are not going to help you with these questions. The only thing that statistics are good for is helping you determine which findings should be interpreted and which findings should not be interpreted. And in some cases, which findings should be interpreted, how much confidence you can have in interpreting certain findings. So I say only, this is not to trivialize the importance of statistics, but I think it's important to realize that in the sort of the bigger scheme of things, in the context of doing science and research and data analysis, statistics is an important part, but it's only one part. It's just one small part. There are many different kinds of statistics. There's t-tests, like what you see here on the screen, and ANOVAs and factor analyses and correlations and all sorts of chi-square tests, all Bayesian statistics, all sorts of different kinds of statistics. What I would like to impress upon you is that all statistics water down to a ratio between signal and noise. So you can think about all of the statistics that you will ever use as being this ratio of signal to noise. And the difference amongst different kinds of statistical measures is how you get to this ratio. So how you define the signal, how you define the noise, and so on. But this ratio of signal to noise is really at the core of all statistical measures. So this may also seem a bit trivial, but this is also important to think about when you are designing your experiment and analyzing your data. There are two ways to get a significant result. Well, I guess three ways. One way is to increase the signal. The second way would be to decrease the noise, so this ratio gets larger. And of course, the third way is ideal. You want to have both a large signal and a small noise. But this is something you should think about even when you're designing your experiment. So if you know that you're going to have, if you know you're looking for a small signal, so let's say a subtle effect, then you want to make sure that you can decrease the amount of noise, for example, by having lots and lots of trials. Conversely, if you know that you're going to have a lot of noise and there's nothing you can do about it, let's say you're doing an EEG study in young children, and you know they're not going to be able to sit still. They're going to produce a lot of, well, the EEG data is just going to be very noisy. There's nothing you can do about it. So you probably want to limit your experiments or your the foci of your analyses to looking for large signals, so large effect sizes. Okay. And so, of course, what you would like to see is that the signal-to-noise ratio is fairly large. And so when you're doing statistics, there is a distribution of statistical values that you would expect under the null hypothesis. We'll talk about this more in the future. And then you have your observed value, and you say, well, if my observed value is greater than some threshold, then we would call that statistically significant. And of course, this observed value would be your t-value or your f-value or your correlation coefficient, or any of this measure of signal to noise. Let's see. I should clarify that when I'm talking about statistics, when I use the term statistics now over the next several videos, I'm not referring to all kinds of statistics. So generally you can use the term statistics to refer to like the statistical characteristics or the features of the data, like the mean or the mode and the variance and skewness and these kinds of statistical characteristics of the data. I'm using this in more of a like I don't even know what the right term is here, but when you're trying to attach a p-value to a particular finding or a significance value to the finding to determine whether something is significant or non-significant. That's what I mean when I'm referring to the statistical value and statistics. So here you have this threshold, and if your statistic value is higher than this threshold, then you would call it statistically significant. If it's lower than this threshold, you would say that it's not statistically significant. So of course, then the next question that therefore is how do you define this threshold, and where exactly do you put this threshold in this distribution of null hypothesis values? And this is a trade-off between type 1 and type 2 errors, which you're probably familiar with. If you set the threshold too low, if it's too lenient, then you're very likely to detect true results, but you're also unfortunately likely to accidentally determine that a null effect is a true finding. So it would be a type 1 error, an alpha error. On the other hand, if your threshold is very high, then you can be more confident that you're not going to misinterpret a null effect as being significant. But on the other hand, you run the risk of calling even true significant or even true effects, you're at the risk of run the risk of calling them non-significant. So that's a bit of a trade-off. And in practice, people set this alpha threshold to be p less than 0.05 or 0.01 or something like that. Again, we will talk about this a little bit more in future videos. There's another important concept in statistics, which is different levels of statistics. So typically in most psychology and cognitive neuroscience experiments, we have two levels of statistics. Level 1 is often called within subject statistics, and level 2 is often called group level statistics. And the level here refers to kind of how close you are to the raw data that are collected, and what the unit of analysis is. So, for example, if you have a typical task or trial-based design, then you would have your level 1 analyses. So level 1 would refer to like the lowest or the first order at which you are collecting data, which would be the single trials. So a level 1 analysis would be a within subject statistics analysis. And if you are, so in typical experiments, you would collect data from, let's say, 15 or 20 or 50 subjects. And then if you average over all the trials within different conditions for each subject, and then you do the statistics across subjects on the trial average, not on the individual trials, now we're talking about level 2 data. So level 2 would be trial averages within each subject, and now at the group level, you're doing your analyses based on these trial averages. Sometimes people call this level 1 thing single trial analyses, although typically you're not analyzing single trials, you're analyzing lots of trials within subjects, so I prefer the term within subject analyses. And of course, you're not limited to these two. You might have level 3, let's say if you're comparing different groups of patients, or maybe patients with different levels of medication, then your level 3 would be so let's say you have a bunch of trials, that would be level 1. Level 2 would be the trial averages within individuals. And then level 3 could be, 3 different groups of, let's say, Parkinson's patients with 3 different types of medication, and then you're comparing the effects of different types of medication, or maybe different doses of medication. I hope that's clear. In case that's not clear enough, here's like a graphical representation of the distinction between performing statistics at the level 1 analysis or within subjects versus level 2 level, which would be group level analyses. So here we have n different subjects, and here each vertical line represents single subject or data from a single trial within each subject. So the kind of standard thing to do in cognitive neuroscience is to average all of these trials together. So this would be like from one condition. Average all these trials together, and then the group level analyses are done on these averages. So by the time you get to the group level statistical analyses, this would be the level 2 analyses, you don't have access to the single trial data anymore. You only have access to the trial averages at the subject level. So this would be level 2. This is a level 2 analysis, a group level analysis. And a level 1 analysis, or a within subjects analysis, this would be where you're performing statistics within each subject individually. So there's a difference. So here would be level 1 analysis for this subject and for this subject and for subject N. Do not confuse within subject and group level analyses. These are very different things, and they tell you different things. I should have put it. I have a slide about this. I should have put it in here, but I'll put it in a future video. I think probably the group analysis video. I'll start with this slide, which shows the distinction between level 1 statistics and level 2 statistics, or I should say the kinds of interpretations that you can make about your data from within subjects analyses versus group level analyses. In general, this is also a fairly trivial point, but most of, or I should say, all the analyses you do generally fall into like these two sort of two kinds. So you want to do some kind of a discrete analysis. You want to know, are these two conditions different? Are 2 different time periods different, or frequency bands different? You're not limited to 2, but the point is that these are discrete comparisons that you're making, or comparisons between discrete sets of data, versus a more continuous-based analysis. So now you're asking the question, is there a continuous relationship between this random variable and this random variable? So let's say, this is like some measure of personality. So each dot is a subject. You want to know, are people with higher extroversion, do they have more brain activity in response to pictures of parties or something like that? So this is more of a continuous analysis and a discrete analysis. I think that's it's fairly straightforward. In many branches of sciences and biological sciences and social sciences, parametric statistics are very often done. Parametric statistics include like ANOVAs and multiple regressions and things like that. you should know that these kinds of parametric statistics rely on a few different assumptions about the data. They assume that your data are randomly drawn from a population with a normal distribution. So the parameters that characterize data have a normal distribution under the null hypothesis. Secondly, that the variance is homogeneous, so the variance is the same wherever you are sampling the data from. And thirdly, the third assumption is that the errors are independent. So different data points that you sample are independent of each other. So of course this happens often. There are many situations in psychology and biology where the assumptions of parametric statistics are met. But we will see in time frequency analyses in the next few videos that these assumptions of parametric statistics are often violated. Sometimes one or two of them, but very often actually all of these assumptions are violated. So either we know that the data are drawn from non-normal distributions, or we simply don't even know what the distributions are under the null hypothesis. For some of these statistics, it just hasn't been worked out, so people don't know what exactly the distributions should look like. Variance has different sizes for different frequencies and different time windows, so variances are inhomogeneous. And for time frequency analyses, the errors are certainly not independent. Neighboring time points and frequency points and electrodes are definitely dependent on each other, very strongly correlated with each other. So very often in time frequency analyses and more advanced analyses of EEG or MEG data, non-parametric statistics are generally preferred. So now I always have this slide and I ask people to tell me what are the assumptions of non-parametric statistics, and people get a bit confused because, there are no, and then I say, well, there are these two, and then I have to qualify it later. So I always say there are these assumptions of non-parametric statistics. These are not formal assumptions of non-parametric statistics, but these are things that you always have to think about when you are performing non-parametric statistics. So the first one is that there exists a null hypothesis, and you can identify what the null hypothesis is. And two, that you have data from which to create a situation that would arise if the null hypothesis were true. This may sound really trivial here, but it actually does get a bit tricky sometimes to think about what is the null hypothesis in the data, and how can I shuffle the data, what can I do to the data to create a situation under the null hypothesis. So it's always you should always think about these 2 points, and it's not always so trivial. Part of what goes into point 2 is just having enough data. If you only have 3 data points, I would say you do not have data to create a situation that would arise under the null hypothesis. So part of point 2 is just making sure you have a reasonable amount of data. This slide illustrates the key difference between parametric and non-parametric statistics. In both cases, you have your observed value, so this would be, a t-value or a correlation coefficient or whatever is the statistic value, your signal-to-noise ratio, that you observe in the actual data. So of course that would be identical whether you're using a parametric or non-parametric test. The key difference is that with parametric statistics, you assume that the parameters, the statistical parameters that characterize the data, have a particular distribution under the null hypothesis, and you don't actually have to compute this distribution. You just assume it. You guess that it is like this, or you assume that it is like this. So for example, it could be a t distribution or an f distribution when I have a longer tail, or chi distribution, chi-squared distribution. There's various kinds of distributions, but you're always making this assumption. You don't actually measure this distribution of parameters under the null hypothesis. You just assume that it's there. With non-parametric statistics, you don't make any assumptions about what the shape looks like. And the idea is that you create an empirical distribution of null hypothesis values based on the actual data that you've collected. So this is where the permutation comes from, or the term permutation in permutation testing. Because you want to permute your data or shuffle your data in a way that you create a situation that could arise if the null hypothesis were true. And then you build an empirical distribution and compare your observed value to this. So in the next lecture, we'll talk more specifically about how you go about creating this null hypothesis distribution. One final point, which I think is also, it's a little bit trivial, but it's important to think about this distinction amongst statistical significance versus theoretical significance versus clinical significance. Statistical significance is really just, if the p-value is less than 0.05 or whatever the threshold is. Any computer can determine whether a finding is statistically significant. You don't need a human being to do this. Theoretical significance is, I don't think we have computers that are really smart enough, or at least this would take a pretty clever, either a pretty clever artificially intelligent system or a really stupid, simplistic theory to determine whether a finding is theoretically significant. So this, we would think of as, requiring some level of human intellect. And clinical significance, of course, if you find that, some substance that manufacturers are putting into our food does not adversely affect health, so there's a non-statistically significant result. Of course, that does have high clinical significance, even with a non-statistically significant result. Well, you get the idea. So this lecture was just a sort of introduction into the next series of lectures, so we make sure that we're all on the same page. And now we get into the nitty-gritty details of how to do, in particular, non-parametric, permutation-based statistical testing on time frequency analyses, time frequency data.